Hello, everyone, and welcome to today's webinar, Spatio-temporal characterization and quantification of DNA replication events by molecular combing. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Genomic Vision. To learn more, visit genomicvision.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Cyndie Forestier, Scientific Account Manager at Genomic Vision. Cyndie, you may now begin your presentation.
So good morning, everyone. My name is Cyndie Forestier, and I am Scientific Account Manager for Genomic Vision. And today, I am going to present you how molecular combing technology with Genomic Vision Replication Combing Assay, RCA, enables spatio-temporal characterization and quantification of DNA replication events for a better understanding of replication kinetics. So first of all, I just want to give you a quick introduction of Genomic Vision. Genomic Vision is a French company based in Paris area at Bagneux, and it's a provider of tools for molecular combing for the detection, visualization, and characterization of mid-range genomic rearrangements or events and their quantification at a single DNA molecule resolution. The company has been founded in 2006, but it really started to shift as a commercial organization from 2019 with the launch of the Fiber Vision system. And since, it has been commercializing different products, instruments, and services to cover a large number of applications with molecular combing technology. So in this presentation, we will cover 4 different chapters. Introducing molecular combing technology first, so what this technology is all about. Then we will see the principle of RCA in single DNA molecule resolution. Then we will give some example of RCA leading towards drug discovery and treatment refining. And finally, we will see Genomic Vision's product and services to enable this workflow for the characterization of replication events. So I'd like to present you first what is molecular combing. So the principle of molecular combing is essentially that we are stretching DNA molecule to resolve it at high resolution and looking at physical and structural variants and replication tracks. We characterize them and quantify this feature at a single DNA molecule level. You can see on the right-hand side of the slide an example of the stretched DNA that is not fluorescent. And molecular combing with fiber technology uses constant stretching factor to provide visualization and quantification of single molecule DNA that can be used to quantify the lengths and distances in genomic features. So really, Genomic Vision has revolutionized molecular combing to visualize quantitative structural features of DNA for a range of applications in single molecule DNA analysis. So the principle of molecular combing is that molecular combing helps to characterize genomics across multiple innovative applications, such as in genetics, in cancer research, genome engineering, and drug development. For the purpose of this presentation, we are going to focus on cancer research application, as the technology gives you accurate measurement of DNA replication kinetics, as you can see on the graph here, with the origin of replication that is pointed with the white arrows and the replicon with labeled in red and in green signals. And in drug development also type of application, as the technology enables us to identify various features of DNA replication stress, but also help us to validate drug candidates through the understanding of the replication events induced by the treatment of the cells with anti-cancer drugs, for example, like PARP inhibitor which is shown into this example here on the picture. So today we are then focusing on the principle of RCA, replication combing assay, at a single DNA molecule resolution. So first of all, we need to understand why replication studies are important. And as we all know, the DNA replication is a crucial biological process by which DNA makes a copy of itself during cell division. During the cell life cycle, the cell goes through different phases, like G1 phase, then DNA replication, followed by mitosis, to then give the two daughter cells with the same genetic materials in each daughter. Since DNA replication happens at a really early stage of the cell cycles, you can imagine that any problem that is occurring at the replication level, whether this is a stop in the replication or a modification of the replication kinetics, will have an important impact on the genome stability, and this has been shown in numerous scientific papers as related to human diseases, including cancer. As you can see in the graph on the left, genome stability mechanism can be at a micro level, so chromosomal integrity is protected by centrosomal pathways in this case, or at a micro level, at the chromatin level, where the DNA damage response mechanism are protecting chromatin from DNA double-strand breaks. In the oncology field, one can study the genome stability mechanism at the micro level by studying replication kinetics through an accurate measurement of DNA replication kinetics, but also the fundamental mechanistic of the replication machinery involving different DNA and damage and response pathways, and the study of the replication inhibitors and activators, but also the replication effect of rearrangement in target genes. This lately gives an indication of the genome stability at the macro level. So comparing the replication events in normal cells versus in cancer cells using our unique replication combing assay leads to drug development, as we can now identify accurately the DNA replication stress events. We can screen the different drugs and test their efficiency, identifying and quantifying the impact on the DNA replication and ultimately onto the genome stability. So for this type of research studies, genomic vision with a replication combing assay offers a clear readout of spatial and temporal characteristics of DNA replication. RCA enables to characterize the replication events throughout the entire genome at a single DNA molecule level. So this is an image that you can see below, generated from our fiber vision scanner using a cover slip, where the DNA samples has been labeled using our RCA protocol and combed using the fiber molecular combing system. You can see in the blue the single-stranded DNA is contained with yo-yo, and the replication tracks in red and in green with the incorporation of CLDU and IDU respectively during the replication. So the arrows are pointing out the origin of replication, so you can already see the power of the technology detecting the replication bubbles with a bidirectional replication forks at a single DNA molecule resolution. So several different parameters can then be assessed with this technology. We can assess the initiation rates, the fork velocity, the fork progression, but we can also combine this technology with fish probes to then analyze activated origin patterns and kinetics at a region of interest. So now this slide walks you through the different steps of the workflow using our replication combing assay in combination with molecular combing platform. During the first step, the DNA samples have to be treated with pulse labeling of replicating cells, enabling the incorporation of two thymidine analog, which in this case are CLDU and IDU, to have a chlorum or iodium that can be used for immunodetection and visualization later on. In the schematic here on the right-hand side, there are three origins of replication that are represented as gray circle. And we know that in the human cells especially, the replication is asynchronous, so not all replications are fired at the same time. Here we are doing the pulse labeling with each analog for 30 minutes. And we start with the first analog being CLDU in red, followed by the IDU in green. So in the first bubble, we can see the replication firing has already started before the incorporation of CLDU. So we can see the origin of replication and the red track. The second bubble shows the incorporation of CLDU during the replication firing, so there is only the red track. And in the third bubble, the cell is not replicating yet during the first pulse, so there is no red track as there is no incorporation of CLDU. After the first pulse, we wash out the CLDU and we do the second pulse with IDU, which then show the green track after the red track for the bubble 1 and the bubble 2. And we can only see the green for the third bubble as the replication firing only occurred during the second pulse labeling with IDU. We can then see the entire replication track with in blue the contact staining of the single-stranded DNA with yo-yo, and the replication tracks in red and in green for CLDU and IDU. We allow the identification of the origin of replication as well as the timing when the replication was firing during the 60 minutes of the two different pulses. By using RCA, we can then have an accurate measurement of each single tracks in order to have precise statistical analysis. So that was for the step 1. In the step 2, after the pulse labeling, we then extract the DNA with a fiber prep kit to extract long DNA molecule. In step 3, we then comb the DNA using our FiberComb molecular combing system. And then we continue in step 4 with the immunodetection step consisting of using fluorescent antibody against the thymidine analogs. We finally finish with the automatic detection and analysis of the replication events via our Fiber Vision Scanner and Fiber Studio software respectively. It is now possible to understand cell viability and specific events only by measuring the replication parameters. RCA is then a powerful tool compared to other technology in the lab, as it can give you several different types of signals depending on the time of origin firing, like we've seen with the bubbles with the 3 bubbles in the previous slides. So in the case of the initiation 1 shows a replicon with in the middle. You will see the replication firing origin contained in blue. Then the replication tracks in red and green either side. That corresponds to the bubble 1 of the previous slide. The second bubble will then show the initiation 2 signal where the replication is fired during the first pulse of CLDU. Therefore, you will see only a red track without any blue in the middle. You will see only a red track with either side the green track for the incorporation of IDU. The initiation 3 represent the bubble 3 from the previous slide, where the replication is fired during the second pulse labeling with IDU. And with molecular combing, you can also detect 3 types of termination signal when the bubble goes back together on the other end. And in case of the first bubble, you would see the termination 1 with the only one red track, as we consider that the green track are fused together as the replication has been resolved. For the second bubble, the green tracks are closing together, then followed by the red track. And this is the termination 2. And then for the third bubble, the termination 3, with the green tracks taking not completely joining. They are not completely joining. They will take time to join, but you will see the, and therefore you will see the blue DNA signal in the middle. In some cases, we can also see the unidirectional fork showing one green and red track, as the other green and red track are so far away on the DNA fiber that is considered as unidirectional. So RCA simplifies to uncover complex replication kinetics that are difficult to reveal since the asynchronous nature of the replication. So the Replication Combing Assay, RCA, addresses various questions about replication on individual DNA fibers in a single experiment. So just to remember, for the DNA analysis, we only take the intact signals and discard the DNA break that could have occurred during the stretching forces of the DNA. And for that reason, it's very important, like I mentioned in the previous slide, that we see the blue signal either side of the replicon in green and red. So the Replication Combing Assay allows us to look at the fork velocity and the progression, as well as the fork stalling by establishing the ratio of signal CLDU-IDU. If we consider that the pulse is the same, it's during the same time for CLDU and IDU, if we see that this ratio is equal to 1, then we will consider that this is an unperturbed replication with the signal in the red is equal to the signal in the green. Now, if we see that this ratio CLDU-IDU is reduced, is less than 1, this will show a slower replication. And in the picture here, we can see that the green track is shorter than the red track, showing that there's a smaller incorporation of IDU. We can also identify with accuracy the length of the replication track, so the replicated DNA on the whole DNA fiber due to the constant stretching factor on our FiberComb molecular combing system. And then we can study the initiation rate, which is effectively the number of activated origin on the total length of DNA molecule. So just to give you an example, for RCA at the beginning of the experiment, we recommend to use 30,000 cells per cover slip. And this generates in average 3,000 signals of replicated DNA on the whole cover slip. So the constant stretching factor is also important for the precise measurement of the forks in order to see the fork symmetry. You can see in the image on the left that the replication track in red and green is longer than the replication track on the other side, showing that there's an asymmetry of the fork. In the case of fork stalling, we can see that the replication track on the right side is lacking the green signal, and this indicates that the replication has stopped. Lastly but not least, as we can identify the origin of replication and measure precisely the length of the DNA fibers, we can also calculate the inter-origin distance and inter-termination distance. So the inter-origin distance being the distance between the different origin firing of replication. This information is actually really important, mainly when we do drug development studies, and we want to check the firing of origin of replication that can be increased after drug treatment with inter-origin distance becoming shorter. So the IOD, we call it inter-origin distance, IOD becoming shorter. And with a panoramic visualization of the replication events at a single DNA molecule level, RCA and molecular combing offer then a really clear readout of spatial and temporal characteristic of DNA replication. So you can already see from the different parameters and the images that we've just seen in the previous slide that RCA with molecular combing gives clear advantages in comparison to classical methods. On each cover slip, long DNA fibers up to 3 megabases will be stretched uniformly and parallel, allowing more data to be exploited than other classical methods, where fibers often cross each other and are then discarded from the analysis. As you can see on the right-hand side, you can see a picture of the parallel DNA fibers obtained with our fiber vision scanner, and versus at the bottom with classical method where the DNA fibers crosses each other. RCA with molecular combing and our fiber vision scanner offer high-resolution data allowing the detection of rare events, and with a constant stretching factor we can determine IOD and ITD as well as Fock symmetry, whereas it is not possible with classical methods like fiber spreading. Fiber vision scanner with also the Fiber Studio software and very soon the integration of artificial intelligence software enable the automatic detection and characterization of the replication events, making the data analysis very robust and standardized and reducing the time to results. So genomic vision with the replication combing assay and the molecular combing technology and platform covers all the steps leading to success from sample preparation to data analysis in 4 different steps. First of all, the sample preparation with RCA and DNA extraction with the fiber prep kits using 30,000 cells per cover slip. Then the DNA combing is done on the FiberComb molecular combing system to stretch the DNA fibers. Then the staining is done and the cover slip are then scanned on the fiber vision scanner for the detection of the signals, and data analysis is done on the Fiber Studio software, resulting in a report for each cover slip and each sample. We then move to the 3rd chapter of this presentation that focuses more on the application of RCA, so Replication Combing Assay, for the use of drug discovery and treatment refining. So when we study DNA damage repair pathways, specific biomarkers have been identified as target for potential anti-cancer treatments. And these biomarkers are involved in different DNA repair pathways, such as non-homologous enjoining, homologous recombination, and other pathways. The most common is PARP inhibitor, so PARP1 pathways, with PARP inhibitor and Pol theta. But there's also other pathways with BRCA1 being one and RAD51. These pathways are using the replication machinery, so the effect of the drug can be monitored and quantified by RCA and molecular combing at a single DNA molecule resolution. So if you want to screen different drugs and study the efficiency of the drug, knowing the target of the drug, there are different ways to design the experiment. So for example, for studying replication kinetics, you can do the drug treatment for the entire experiment, even during the 2 pulse labeling, or in between the 2 pulse, and then look at the fork progression. In this case, if you do the treatment after the 1st pulse, you can see the progression of the green track for treated versus untreated cells, and see a shorter track on the right-hand side indicating a slower replication. Or if you focus on restart of the replication studies, in this case, the drug is administered during the 1st pulse, then washed away with something like hydroxyurea, or you can do the drug treatment in between the 2 pulses for a period of time, then wash away the drug. You can see the green track shorter than the red track as the replication restarted, or you can see stalled fork with only the red track, or new origin with only the green track. And finally, you can also do resection studies where the drug will bring you to the reverse fork, and in this case, the cells are treated with the drug after the 2 pulses. If there is resection, the exonuclease activity of the replication machinery will take off the DNTPs of the IDU track, and you will see the green track shorter than the red track. This is applicable for many biomarkers, many drugs like PARP inhibitor and if there is resistance to the PARP, the resection studies may be analyzed or can be checked on the new firing origin. And this is the case for all biomarkers being anti-cancer drugs. So this is an example of the use of molecular combing technology with RCA for new anti-cancer therapeutic strategy. BRCA1, 2, and RAD51 play a major role in replication fork stabilization following the replication stress by preventing nucleolytic degradation of replication forks by the nuclease MRE11. And in this paper, Dr. Sanij and her team wanted to investigate the effect of CX5461 on fork stabilization in OVCR8 cells. So nascent replication tracks were first labeled with CLDU, then IDU, before treatment with CX5461 for 3 hours. They then looked at the replication fork progression by measuring the IDU track's length. And we can see on the graph here that with the CX5461, the IDU track is shorter, indicating that CX5461 treatment causes an overall decrease in track length, as you can see in the graph also with a red plot. This suggests the degradation of replication forks upon induction of DNA damage and repair by CX5461. And this was rescued by co-treatment with the MRE11 inhibitor, which is called mirin, confirming the inhibition of the MRE11 nuclease can rescue CX5461 mediated fork destabilization, as you can see in the plot in green and in blue. In the second graph, so on the right, they then assessed whether DNA damage induced by CX5461 treatment affects fork progression by pre-treating cells with CX5461 for 24 hours, then post-labeled with both analogs. Pre-treatment with CX5461 had no effect on the fork length, suggesting that CX5461 does not cause any lesion that could impact fork restarting or progression. As you can see in the red plot, that is no different from the control plot in the gray. On the other hand, the PARP inhibitor, so talazoparib, which is called in this case BMN 673, increased fork progression. And this was confirming that PARP inhibitor mediated acceleration of fork elongation is a mechanism for replication stress and DNA damage. As you can see with the BMN 673 plot in green, and later on with the blue plot with CX5461. So to conduct these experiments, Genomic Visions offers an end-to-end solution with product and services so that you can have the choice to either conduct the experiment in your lab at your own speed or simply send the plugs, the samples to Genomic Vision for servicing. So there are 3 types of consumables that are required for the sample preparation. The fiber prep kit is used for the extraction step and the preparation of the plugs. So it ensures high molecular weight DNA from 100 kb to 2 to 3 megabases. And you can use it with whole blood cells or pelleted cells from cell culture. There are 2 formats for this kit, so for 10 samples or 400 samples. And depending on the format of the kit that you will go for, the storage and shipment will be different. So for example, for the fiber prep kit 10, this is shipped at 4 degrees, and for the fiber prep kit 100, there will be 1 part shipped with dry ice, which will then need to be stored at -20, and the other part at 4 degrees. Then for DNA combing step, you will then need to use a single single-use disposable reservoirs that fits the FiberComb molecular combing systems. And these reservoirs are single-use, sterile, and DNA free. Then finally, the engraved cover slips are strictly quality checked and make sure that DNA binds from the extremities and give reproducibility and robustness to the combing of the DNA and DNA affinity. For the combing of the DNA onto the cover slip, like I mentioned, we have our FiberComb molecular combing system. And this system allows the stretching of the entire genome uniformly and continuously in a 10-minute procedure. So the advantage of this system is really that DNA molecule size are between 100 kb and 2 to 3 megabases, which is very long for DNA fibers. But it's mainly the fact that it allows parallel alignment of single DNA molecule due to the constant stretching factor. And DNA extremities are not chemically modified. So for the scan of the comb cover slip, you can use either the FiberVision or the FiberVision S scanner. Both scanners have the same high resolution at 0.16 micron per pixel and can allow a quick automated scan of 60 minutes per cover slip. Now, the main difference between the two scanners, it's the sample storage and the channels. As you can see, the FiberVision allows 160 sample storage with permanent loading, and there is 3 channels for simultaneous 3-color labeling. Now, the FiberVision S offers a more flexibility and versatility of the applications, as it has 9 channels colors. But the slide storage is a bit less, with a 12 slide storage and automatic slide loading. So for the data analysis part, we use our Fiber Studio analysis software, and this software allows the detection, analysis, and interpretation of hybridization signals on comb DNA in a streamlined computer-assisted process. Essentially, you can see a photo of the software on the right-hand side, and you can see each of the events is boxed in. Then you can visualize the events, so the replicon in red and in green on the right-hand side, and then you can validate this event. So there's a possible manual correction of automatic measurements. At the end of the analysis, there's an automated report creation and downloadable raw data. And the sensitivity of the software, it's so high that it enables the detection of unexpected features. And it's available for structural variation and DNA replication analysis. Now, to complete this software, Genomic Vision is also launching very soon a new software with artificial intelligence, which will completely characterize all the events automatically and generate directly all the report without having to manually check each of the events to validate the characterization of these events. So now, Genomic Vision offers also the possibility to send out your samples to our service in-house. So for that, you can prepare your samples with a fiber prep kit to prepare the plugs of agarose and send these plugs to our service for the extraction, combing, hybridization, and image acquisition with the EasyComb service. And then the team will send you the raw data to analyze on your site. Otherwise, you can also incorporate the analysis part by choosing the EasyComb Plus service. Now, if you have already got the FiberComb molecular combing system in your lab, you can do the preparation of the samples and comb the DNA onto the cover slips and send us a combed cover slip for servicing by choosing the Easy Scan, and then you can carry on the data analysis on your side. Or if you want to have the reporting as well as the scanning, then you can choose also the EasyScan Plus service. There are numerous scientific papers that have been published showing the use of fiber vision platform with RCA to better understand the events of DNA replication in different pathways and how they are impacted after cells treatment with different drugs. Here you can see an example of 5 different papers. DNA fiber analysis Mind the Gap has been published in 2017. The other ones are more recent with the two last papers, Scientific Report and Molecular Cells, that have been published last year. So as a summary, molecular combing with the fiber vision platform and RCA provides an easy and complete solution to understand the replication kinetics. This technology is the only tool enabling to better understand the efficiency of the replication by various parameters such as replication speed, fork symmetry, inter-origin distance IOD, intertermination distance ITD, and the number of replication. Other classical methods would be compromised in some of these parameters. RCA can be combined with various compounds against specific biomarkers to better discriminate the efficacy on a specific pathways. And it's also an external biomarker for understanding and quantifying the possible drug target and its efficiency. And in all of this, Fiber Studio and our Fiber Vision platform allows to detect all these parameters and to analyze the data with more efficient and standardized quantification. And it's also allowing a faster data analysis, reducing the time to results. Thank you very much for your attention, and I will take now any questions. Thank you.
Thank you, Cyndie, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the number of cells required for the experiment?
So for replication combing assay, we normally use 30,000 cells at the beginning of the experiment. And this leads us to generate, in average, between 5 and 15,000 signals per cover slip.
Okay, thank you. Next question. Why in the report do we get only the initiation 1, termination 3, and unidirectional events?
So the simple reason to that is that we need to border actually the replicon with the blue signal for the single-stranded DNA. In the case of initiation 2 and termination 2, for example, where the firing of the replication is happening during the second pulse, during the first pulse, we will not be able to distinguish the fork asymmetry. So we will be able to give only the extremity colors for the fork speed, but not the fork asymmetry. So this will be not reliable. And this is why we only use replication initiation 1 and termination 3, as well as unidirectional signals for the reports generated at the end.
Thank you. Our next question is, how many signals can be detected on a cover slip using RCA and 30,000 cells?
So it's quite variable depending on to the cell type that you are using, and depending on the density of the fibers, obviously. But in general, this is between 5 and 15,000 signal per cover slip.
All right. And our last question is, which brands of AB do you use for CLDU and IDU? We found none that are truly specific since the original brand was discontinued in 2017.
Yeah, so since a few years now, we use other brands to replace the brand that has been discontinued. And for the CLDU, we use the AppCam, and for the IDU, we use BD Bioscience as a brand for the antibody.
Well, great. Thank you again, Cyndie, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Genomic Vision, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today, and those submitted during the on demand period, will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.
Thank you. Thank you to all, and goodbye.
Bye. Thank you.